My presentation will be focusing more on different omics, and I'm really honored to be here to show you our work on the molecular human. And as Gordon was mentioning, many of us which are here are working together on this effort. It's like multi-center work, and I'm just happy to be here and I have the honor to present you this work. So, human body and pathological as well as pathophysiological processes can be described with different strategies, and the clinical chemistry will be the very first of them. But obviously it's not having such rich information which could comment on the complexity and the architecture of human diseases. That's the reason where further development came and multiple omics arrived, including glycomics, the epigenomics, transcriptomics, proteomics, glycomics, and also metabolomics. And there are many studies which are in fact using all of those in the standard, responding to standard questions, but also in the biobank. But in our view, there are multiple things which are still remain open and need further examination. For instance, what is the complementarity between those platforms? It's very difficult to understand how different omics layers are complementary, right? The other thing is how to integrate and analyze such a huge data set. And the most important, in my view at least, how to visualize them, right? If I were gonna show you the Excel table, it's not really cool to look at the p-values. So it is very difficult to answer because you will need a cohort of the individuals which all were measured for all those omics. And the cohort which I like to introduce you today to is the Qatar metabolomics study on diabetes. And Karsten Sure was the person who was initiating this study together with Dennis and Dr. Salim. And we were able to collect around 390 subjects, including diabetes and controls. And we collected urine, saliva, and plasma from all of those individuals, right? We aliquoted them to start to play with the data. So what you can see here are the all omics which we were generating over the years, because project started around 2013, and right now we are in 2020, right? So we look on the clinical chemistry, we also check on the genetic variants, methylation, gene expression profiling with RNA-seq, microRNA, proteins with two different platforms, which was Olink and Soma, glycans, total and plasma glycans, and also EGGs with Gordon, but also EGAs with Manfred, which is sitting there. We look for lipoproteins using Nightingale platform, lipidomics, two different platforms, including Biocrates and also lipodizer from Metabolon. And saliva, urine, and blood were screened for metabolomics. So it was untargeted profiling using for blood two different generations of metabolome platform, which was HD2 and HD4. Urine was screened for metabolomics with genomics and also metabolome. So differences is different strategies, genomics based on NMR. And also saliva, which was, we use standard metabolome platform for this one. So we exhausted pretty intensively those data sets. And here what you can see is the number of papers which we were able to generate. I'd like to just showcase one of those, which is describing the interaction between plasma and glycome and proteome, which I feel might be interesting for you as a community. So now we have all those 18 platforms, which are technologically different. What I would like to point you here is some of the platforms are describing the same omics, like for instance, Soma and Olink, or glycans, the EGA and EGG are overlapping with some of the molecules, because by Manfred platform we also look for EGJs, also lipidomics and stuff. So in total, we generated information on over 6,000 molecules, which are not on genetic components, together with genetic information on over 1 million of SNPs, 50,000 transcripts, and almost 500,000 of methylation sites. So I feel I convinced you that we were in position to start to answer the questions on the complementarity and start to think how we can visualize those data. And this was our approach. So what you can see here, we started with the mutual best hits. And this is hypothesis-free approach, where you can look for the molecular orthologs. We also applied Gaussian graphical models, which is enabling to infer the interaction between the molecules. In addition, we conducted association between Glycome epigenome and also transcriptome and genome. And in addition, we did multi-omics GWAS, so genome-wide association study, but also EWAS and TWAS. Next, we asked the question about the moleculars which are associating with phenotype which we had in our data, which was sex, age, type 2 diabetes, and BMI. So now we had all of those calculations ready and in place. So what we did next, we tried to check how we can connect those omics. And this is the reason why we developed a server, connecting omics server, which we are calling Comics, which we also fitted with GWAS catalog in order to put it in the context of the diseases which are of genetic relevance diseases. So I would like to encourage you to go and check our server, and I will show you how does it work. I hope you will gonna have fun as much as we are having every time we are trying to generate some hypothesis. It will be freely available, and I hope you will gonna have a chance to look at it soon. So what you can see here is our server. First of all the data and all the statistical summary is here. So you can download and play on your own wherever you want. Then let's go back to the server. So here are all our platforms which we use, so you can check for the platforms, but also you can check for molecule of your interest. And let's say we can do it even here. If I'm interested in glucose, I'm generating the molecular network around this glucose, and I can increase the network, so up to even 200 nodes. And I can understand what kind of molecules are associated with my glucose, with different contexts, right? This data can be also downloaded by the export here, okay? But I would like to bring you back to the presentation. What you can see here is top 20 molecules showing significant association with age, and I would like to highlight that 9 of them were glycans. And I'm not showing it as new findings because everyone here is of course aware of glycan age. I just like to show that our data is really solid. So this is the reason why I'm pointing it here. Right now I would like to bring your attention back to our mutual best hit, and we were using it to answer, first of all, the questions not only of the complementarity, but robustness of the platform. Because sometimes there are issues with annotation of certain molecules. And the first things which we did, we checked for those mutual best hit to check whether the hits are showing exactly identical molecules. So what you can see here are all the platforms which we were using. The lighter color part of this graphic is indicating the number of the overlapping samples. And here, the darker color is showing you the mutual best hits. So the highest number of mutual best hits were between 2 platforms of the metabolome, 2 different generations, which was 369. This was expected. We were checking if those 2 platforms are generating data which is of good robustness, and indeed, mutual best hits were pointing towards exactly the same molecules, and we did exactly the same exercise for all the others platforms which were describing the same molecules within the same omics, okay? So our data is really in pretty good shape, and the technology behind, right? So the next question which we asked was, can we define which platform is outcompeting other? And this we did by looking the strength of the genetic variants. And what I'm showing you here is the glycine measured at 3 different platforms. So you can see here, this is the p-values reflecting on the strength of association between the genetic variant and this particular glycine molecule. And based on this graphic, we can conclude that BM, which is Biocrates platform, performed the best out of others. But if we look on the other example, measuring other metabolite, like for instance here butyryl carnitine, we are seeing that Biocrates platform is not the best for this particular molecule, right? And we check whether we can make any general conclusion which platform is better. But when we look over all the omics and over all the platforms, in fact, we cannot. So it brought us to the conclusion that none of the platforms is out-competing the other. And some platforms are good for some specific molecules, and it will be interesting if somebody is interested in specific molecule set to look at this data. So now something for Peggy. I think this is the mutual best hit was showing us also, or shedding the light on the kidney function. So what you can see here are biological associations driven for those mutual best hits. And what you can see here, you are seeing the association between creatinine and also insulin-like growth factor binding protein 6. Both are known to be involved in the as kidney function, especially the EGF-BP6 was identified as upregulated in children with kidney failure. And those molecules are linking with another metabolite, which is also a molecule suggesting the compromised kidney function. So with this example, I'm kind of emphasizing the complementarity and the biological functionality, which is described by all those platforms, which can be linked together by, for instance, mutual best hit. Now I would like to move quickly to GWAS, because I don't know how we are standing with time. So we are doing GWAS, EWAS, and TWAS, and get a lot of the hits. I would like to say that majority of them were able to replicate based on the previous study, but here I would like to show you something which was new. And what we found those 4 genetic variants of ST3-GAL1. I believe everybody knows that this is the molecule which is conducting the final glycosylation of glycoproteins and also glycolipids, and is involved in cancer and is very important in breast cancer progression. What we found here, we found that each of these variants is possessing unique association with specific glycan structures, which you can see here. And important here is that the glycans which we identified as associated with this variants were all EGAs, okay? So it might be important in context of cancer. We also look for the overlap between them. We find that the only one genetic variant is having a huge number of those glycans which are associated, and some of them, they are overlapping. We check for the directionality. So our question was whether the genetic variance is showing same directionality, so positively or negatively associated, and you can see that different variants are showing different associations, okay? And I believe we are extending on the knowledge on this particular molecule in terms of functionality, which of course should be further followed up in context of, for example, cancer. So now I would like to change a little bit gear and point you towards the functionality of our server. You can look there not only for the different molecules, but also different disease. And what you can see here is the cluster around type 2 diabetes. We saw there 129 molecules which are known to be involved in diabetes, and this serves us, in fact, as such a positive control for the functionality of our server. You can always further ask questions, what about specific pathways which are dysregulated in type 2 diabetes? And here, for instance, you have HbA1c, which can be used as a proxy of glucose metabolism, and you can further explore the pathways around. So the final example I would like to point you here is, again, a genetic variant around FUT6. And FUT6 is known biomarker, and this is, again, a network which was generated with our server. So FUT3 is associating here with 2 different methylation sites. One, this one here, was reported in context of cancer, the other one not, and might be potential novel findings. The variance in FUT6 was also showing association with proteins of FUT3 and 5, which were measured on SOMA, which again are known to be involved in cancer progression, metastasis as well. So when we were expanding the molecular network a little bit more, what you can see here is that one of the methylation site is strongly associated with different glycans, and this could be example of epigenetically regulated glycosylations. And given that this particular methylation site was not previously reported in context of cancer, it's also giving us a specific hint and can kind of support us in further discoveries related to this methylation site. So with this example, I would like to finish my talk. I hope that I encourage you to visit our server. Please have a look, enjoy as we enjoyed working on this project. This brings me also to conclusions. I hope that in this short period of time where I just only touch upon some of the results of ours, I was able to show you that we could look at the complementarity, we can address also integration of the data, but also visualization, which I see as a main problem with the current era of multi-omics. And with all the molecular interaction which we were calculating, I feel we illustrated a bit the molecular human. So we see our project as a benchmark, but also as a prototype for bigger studies, like for instance, UK Biobank and other biobanks which might join multi-omics wave. And we hope that it will be used by the community, and we are very happy to share it. So this work will not be possible with all people which are here, especially Karsten Sure, who is the main driver of this project and excellent mentor. I would like to also thank to our collaborators from Weill Cornell in Qatar and New York, but also our friends and collaborators from Europe, from Munich, from Greifswald and Leiden University, and our collaborators from Croatia, from Genos, for the support with glycans. Thank you very much for your attention, and yeah, thank you so much.